All right, so the next one, so that was base substitutions. The next thing that we're going to look at is adding or removing nucleotides. And the best real-world example that I can give you guys that you probably experience, and you can understand the magnitude of this screw-up, is scantrons, right? Filling out scantrons. If you've ever taken a test, right, and you like skip one because you want to come back to it, but you don't skip it on the scantron, you know what I'm talking about? What happens if you don't recognize that you did that? You pretty much get the rest of the test wrong, right? It screws up the whole rest of the test because you've bubbled in the wrong one corresponding to the test. Do you understand what I'm talking about? Okay. All right. So same thing here, right? This code lining up matters. So if you, in this case, what if, say, right, and teachers would do this crap, right? And in our defense, we would never do it on purpose, not necessarily. But you'd have like 4 A's in a row as the answer, right? So maybe you do 5, like you get carried away and you fill in 5 A's, right? So that's like adding an extra nucleotide here. So what does that do? Again, you have that same problem as if you skipped, right? You just shifted everything. All your answers are not going to line up. Still with me? So here, look at the effect that it has on reading this DNA. So look, you added an extra T here. This is where the change happens. So anything before it is fine, right? The arginine is fine. But notice that this change now results in a different amino acid, not only right here where the change happened, but in the next position. And even worse, in this third position, you're doing what? Stop. So this type of mutation where you add or delete nucleotides and you shift the reading frame is referred to as a frame shift. And it most often results in premature stop codons. So if you were to compare this to a base substitution, which would you vote for? Base, because most likely it wouldn't be a bad mutation, right? But this frame shift, not usually good, right? A lot of change and usually stopping before you're supposed to. So this we're just going to, like I said, I'm going to fast forward over. So here's your DNA. This is how it's supposed to be, right? Here it adds in an extra C. So when you then transcribe that into the transcript messenger RNA and then translate it with the ribosome into the amino acid, this is what it's supposed to be: arginine, tyrosine, valine, lysine. Instead now, with that extra C, it goes to glycine and stop codon. So that's adding. Do you think deleting would have the same effect? You going to get a shift? You going to maybe stop before you're supposed to? Probably, right? So here's a real example, right? They forgot one. This is supposed to be valine, now it's leucine, aspartame, right? I don't show you the whole thing, but chances are you're probably going to stop before you're supposed to. So in trying to explain this to my students, ignore the poor grammar, okay? Like my grammar's not right. But the cat ate the dog. These are all 3-letter words, right? And you can read it despite the crappy grammar, right? It makes sense to you. Right? Okay. What'd I do to that sentence? I removed the T, but I did just like a ribosome is programmed to do. 3, 3, 3, 3. So I grouped the letters in groups of 3 in codons because that's what a ribosome would do, right, if you gave it code. It can't think. It just reads, right? So you give this to some poor kid, right, and he's going to try and pronounce this. That's what we call nonsense words, right? And exactly, it makes no sense, right, to us. And it would be all screwed up for the ribosome as well. So that's removing what I do here. I added, and again, look, we ended up with a real word, T. But again, is that going to make sense to anybody? You give this to a poor kid, right? You know, and if he was trained like a ribosome, 3 letters is a word, he'd be having a time. Now, there's one exception to this where you won't get a frame shift. Okay? Adding and removing nucleotides. So in this sense, where could I add the word big to help make this sentence make more sense? Well, obviously, the cat ate the dog. Well, maybe it was a big cat, right? So the big cat ate the dog. I inserted a word. And remember here, what are the words representing in terms of genetic information? What am I using this as an analogy for? What are these words representing as far as DNA and RNA goes? Codons, right? They're representing codons, because remember, codons are 3 nucleotides. Did I cause a frame shift by adding this codon, this word? No, right? Everything's still intact. It makes sense. But did I change the code? Yeah, definitely, right? In terms of phenotype, what did I add? So I added a codon, but in terms of phenotype, what did I add? What do codons code for? Amino acids. So I just added an amino acid, right? So let's actually look at it in DNA and RNA code. So here's the wild type, right? DNA, because you see what? T's. RNA, because you see U's. And then these are the 3-letter abbreviations for what? Amino acids. So this is how it's supposed to be. What's different down here? What I add at the bottom. Yeah, there's 2 values, right? Is the first one supposed to be there? No. Do you see that? Right there? That's new. That's new. That's new. But the rest, it stayed the same, right? There's no frame shift because it's the same amino acids. So again, what effect would that have on a protein, say the enzyme lactase, if we added an amino acid? What do you think? Does it become super lactase? It could. Does it become, I can't do it anymore, lactase? I don't know how to grab onto lactose. Maybe. Does it affect it at all? Could have no effect, right? It could be any one of those outcomes, right? It could have no effect. Could make it super good. Could make it super crappy. Could have no effect. Could run the gamut. But what you don't have here that really usually is really bad is that shift, right? So this is the one exception when you add a whole codon, 3 nucleotides, or even remove one, right? We could remove, that you don't get a frame shift. But it has to be in between codons, right? So we notice we added it here. But if I added CAC, like right behind this T, then you'd get a shift, right? So this is rare. But it is possible, so we do have to talk about it. So who discovered transposons? I said it earlier. It's a woman. Barbara McClintock. I did actually meet her once at a meeting. The American Society for Microbiologists met in Boston right before the conference that I went to. And so a lot of people were coming directly from that. And next year it's going to be in New Orleans. So there's already a contest to determine who's from the workshop is going to stay at my house. They come to New Orleans next year. I'm excited that I don't have to travel to the meeting, except now I'm trying to figure out how I'm going to pay for it. Because it's not free. You know, scholarships, fellowships, begging. Somehow I'll go to the national conference, so I don't have to fly. But sometimes you have really amazing people like her that come and talk at these things. And kind of like the beginning scientists we talk about, like Anthony von Leeuwenhoek and stuff, you know, I mean, they just kept asking questions. Like, you know, they saw a phenomenon and they're like, how did this happen, right? You know, what is going on here? How can I explain this? Or in the case of Anthony, was like, wow, look at this. I want to tell everybody about it, you know, what I'm seeing. The sad thing, though, is because he wasn't, well, not to say that all scientists are people that will share their stuff. He would not share his lenses or how he made them with anybody, which really kind of sucks because from what they said, his were pretty impressive. I mean, he saw stuff, pretty impressive stuff. Like, he made some pretty insane lenses, but he didn't share that with anyone. He shared what he saw with people. He told them he did drawings or had drawings rendered and wrote letters to the Royal Society of London. But he didn't share his materials or his methods, which is unfortunate. I, on the other hand, probably overshare. I apologize for that now. So she saw this phenomenon, right? And she discovered that segments of DNA known as transposons, because this isn't normal, that could spontaneously move from gene to gene. So remember, gene is a segment of DNA that codes for something. When they jumped from one spot to the other, they would disrupt information that's already there. So I was trying to think of a real world example. It's almost like, I don't know if you guys remember in the old days, of course that's old for me but not for you, party lines. Like when you would use the phone, someone else would already be on that phone line. So you go to talk and someone else is already talking, right? So like your information gets all screwed up. You do know what I'm talking about? Or probably never experienced it, but you've heard of it, right? Okay, good. So, or like on the radio, sometimes you'll get two stations at the same time. That really happens nowadays, right? So, you know, you really don't get what they're saying because there's two things of information overlapping. So in this case, imagine, you know, a gene with another gene in it. How is the system going to read that? Well, one of them it's not going to get, right? Because it's disrupted. It's only going to read what's coming through clearly and is not disrupted. Still with me? Okay. So transposons disrupt, so therefore the proper function of a gene. When they jump into that gene, and that's another name that's sometimes given for transposons, is jumping genes, because they do that. They jump from one segment of the chromosome to another. They induce, we can do this in the lab. It's happened in nature. She discovered in nature. But we can cause this to happen on purpose in the lab. Like we understand the mechanics of it. And we can make it happen. So organisms have had this done to them at the organismal level. So one of the researchers I was talking about at the workshop, she was saying about how it didn't work in one of the mouse lines that she was working with. When I was at LSU doing research, there was an immunologist working there. She had a specific line of mice that had been specifically bred and genetically modified that they had a specific gene knocked out, right? So where that gene was disrupted so that they no longer made that gene product. Like they couldn't make that product, whatever it was. I wish I could remember exactly what it was, but I don't. But I knew she had knockout mice, and I remember people saying that, and I was like, what? There's lots of different knockout mice out there. What that particular researcher had at the time, I don't know. I mean, that was like 20 years ago for me now. 15. Too long. And it wasn't my research. It was someone else, you know, so it wasn't personal to me. But again, they kept saying, you know, her knockout mice, and I'm like, what the hell are they talking about, knockout mice? I'm like, oh, yeah, it doesn't produce whatever, you know? And I'm like, oh, why did they do that? Why do you think they would purposely do that? Like have a mouse not be able to do something. When you have disease, right, you can't do something, right? So they purposely made it so it couldn't do that so that they could study it. Right? Does that make sense to you guys? So this is used a lot in research where we can make mice be like the disease so we can study the disease or the defect or even understand some component of how the system works. Well, what if you don't have this? What happens? So sometimes it's also referred to as an insertion mutation because, again, you're putting something in order for something to not work. So this was my best example at the time and something that I did for real in research. We would use these specially designed circular pieces of DNA in bacteria known as plasmids. And they were designed in a particular way that you could insert genetic information into them. And the reason why we did that was that bacteria are really cheap copiers, and especially when they're like cheap copy machines when it comes to DNA, right? And when it comes to DNA, like you can do polymerase chain reaction, in a thermocycler, but you're limited as to how many base pairs. I forget how many you can do in that. But if you put it in an organism like a bacteria, they'll copy huge amounts of DNA for you. And then you just, kill them and steal it. We just pop the cells and take back the DNA they copied for us. And it was designed in a way, too, that, we could do that. We could take those and purify out those pieces of DNA, those plasmids, and then we could send them to be sequenced. Or in my case, I actually sequenced them in the lab that I worked in. Actually loaded the big, huge sequencing gel. The fancy pipettes. But in order to know, right, that it actually worked, like that I got the DNA in that plasmid and the cell was copying it, the plasmid was designed in such a way that we could tell that they had done it without having to purify and run it on a sequencer and then go, oh crap, I have nothing. Right? That's a lot of work to be like, oh, it didn't work. Because does it always work? No, right? There's a percentage and things like that. So this particular plasmid or construct that we worked with had a segment that was designed for our stuff to insert in. And this particular segment coded for an enzyme that would split a molecule, a substrate that we gave it. And so when they produced the enzyme, they would split it, and it would go from colorless to being blue, right? So this substance we gave them was colorless. When they produced the enzyme, they split it, and they would literally, the E. coli colonies would be blue, right? So that's in their state that we have them in. If we inserted our thing, then hopefully they no longer produce that enzyme and they no longer could turn blue, right? So you would have ones that it inserted into. So here is that gene, right, in gray. And here is maybe our sequence we inserted in. So now they're not making the enzyme anymore because the enzymes, right, the RNA polymerase isn't going to jump from here to here and put these two pieces together. It doesn't work that way, right? He just follows along from start to stop, right? And here he's starting, but then he's hitting another start, so he does just that. He starts over, right? And he'll just copy this information, and he won't make this anymore, right? Because he only takes a piece of it. And so that's not enough. So they can't make the enzyme, they can't turn blue anymore. But for us, that's just not enough. We've got to be selective too. So in addition to our sequence, we would actually give them antibiotic resistance. We would give them the ability to make an enzyme to destroy an antibiotic, say penicillin. It wasn't penicillin, but everybody knows that penicillin is an antibiotic, so that's why I wrote that. Make sense? So my plates would contain an antibiotic, right? And only the bacteria that my insertion worked for, right, that they got this gene and my code, would grow on these plates, and they would be white and not blue. Still with me? Pretty cool, huh? Right, so I would grow them up in large numbers, lyse them, grab the DNA, sequence it. So one example of how, and then like I said, the other example where they literally have done that at the organism level, that mouse does not, its DNA is disrupted, it doesn't make a particular protein anymore, it doesn't have a particular ability anymore. The whole mouse knocked out. So there's Barbara, and she actually discovered this phenomenon in maize corn, and when I would say that, my students didn't know what I was talking about, so I found a picture on the internet. So this is where each one of these corn kernels, y'all, is a baby corn. It's an embryo, right? It was an egg that was fertilized by another. And so each one is different, but yet usually not very different. So it's all pretty much from the same plant, right? So all of them should be the same, right? And they were pollinated by the same pollen, so why aren't they all the same color? Well, genetically she found that they really were pretty much the same. The problem was what genes were disrupted and what ones were not. So some genes were disrupted that produced the different colors. So you had this jumping, right, effect. So here again, when you look, so say you have these genes, 1, 2, and 3. They jump into B, right? So they like separate B like you saw before. So now they can still do A and C and 1, 2, and 3, but B they no longer make. Well, maybe that is why this one is white. Because of that insertion, deleted B, which was maybe the color yellow. So now they're white instead. Make sense? So this animation is kind of good and kind of bad because it shows you stuff moving, but it doesn't really show you the disruption, right? So I mean, these things can move all over the place, right? They can go from a plasmid, right, and jump onto the chromosome, off the plasmid, and then one from the chromosome can jump to another spot of the chromosome, and that one can jump there, and it can jump onto a plasmid and leave a cell. But it really isn't showing you what really happens is that each time they're jumping in, they're probably disrupting a gene, right, and affecting the function of that gene. So they're essential, right? So why would a scientist do that? Why would a scientist knock out a gene? Well, because then that helps us understand how and why and what these things do, right? So inducing mutation, actually causing changes at the DNA level, helps our understanding of genetics, right? How things work. When we do this on purpose, they're referred to as induced. So even though transposition, transposons, right, this phenomenon of insertion, knockout, happened in nature, right, and Barbara observed it in the maize corn, we can cause it to happen in the lab. Chemical mutants, right, this is, you know, some people are like, well, it's natural or it's not. But sometimes when we purposely take a chemical, right, that we know will mutate, and we expose organisms to it, it's induced. Right? Although you may be exposed to these things in the natural environment, right, from some other organism or something like that. But typically when we're purposely exposing, this is referred to as an induced mutation. And again, radiation, as we said, you're naturally exposed to it. So you have a natural level of mutation that's spontaneous, that's induced by radiation. But when we like purposely blast something with additional, right, we're purposely doing this, we're purposely trying to induce this. So in laboratories, in biological safety hoods that some scientists work in, that create a curtain of air to create a sterile environment inside that hood, that is meant to protect the person working with the stuff and also to keep the stuff they're working with from getting contaminated. Most of the time it's to keep us from getting contaminated. That's why you would work in a biological safety hood, which I did when I worked with leprosy because, you know, I don't want to inhale it and get leprosy. So when we weren't working in the hood though, we turned off the fluorescent light and we turned on the UV light, right? Because UV radiation, as we know, impedes microbial growth. Right? You wouldn't want to work in there with it on though, right? So we turn it off and we turn on the regular light when we're working in it. But to help keep the inside free of microbes, we would use UV light. They're using UV light more and more now to quote unquote disinfect water. The only problem is that UV light isn't very penetrating, right? That's why you can put something like sunscreen and be protected, right? Because a very thin film like sunscreen will protect you from UV radiation. So it doesn't penetrate very far. So you have to run very thin ribbons of water past the UV light in order for it to kill any of the microbes in the water. But it's being used more and more to disinfect things like water and air. So another thing that when you work, especially in the biotech industry, one of the most common chemicals we would use in the lab is aethanium bromide. And the reason why we use this substance was it's in the class of chemical mutagens known as intercalating agents. So what it does is it binds in between the bases to DNA. But the reason why we use it in the lab is that when it binds to the DNA and you expose it to UV light, it fluoresces. So you see these bands, these lines in this gel? So DNA ran through this gel matrix with an electrical current, and it moves based on the size of the DNA. So here's a standard. We know the length in base pairs of each one of these pieces of DNA, and this would be an unknown sample. So we would run it out so they separate out into the different pieces, and then you'd put it on the UV box, and then you would be able to take a picture and see where your unknowns line up with the known ladder. And so I have a picture of that too from my workshop too. Should add my new picture in here. But we didn't use aethanium bromide. Why not? It's an intercalating agent. It binds to DNA. So if it binds to your DNA, when your cell goes to copy the DNA, remember spacing is important, right? So here we have, right, a spiral staircase where instead of stepping up 2 feet, you got to make a big leap because there's a missing step there apparently, because that intercalating agent's there. And so now you've got to hop up 4 feet. I don't know about you, I'd be calling the carpenter back and be like, dude, put another step in there. So what does your DNA polymerase do? There's a gap, right? So what does he do? No, he might stop, or he might add another step, right? Add another nucleotide. Okay. So, is that good, bad? What could happen? Frame shift, right? You're going to get an addition, probably, and you get a frame shift. Now this DNA, right, this isn't in a body though, right? We took it out, it was in a test tube, we're putting it in a gel, right? No problem, right? But the scientists working with this, what would they do to protect their DNA? They'd wear gloves, for sure, right? The bottle of aethenium bromide in the lab that I worked in had a skull and crossbones on it, which stands for what? Yeah, death. But in this case, mutation, possible carcinogen, right? Because it will mutate your DNA. So in your body, this happens, right? So if you're not wearing gloves, your skin cells get exposed. There's one of the teachers that teaches on the West Bank. He worked in the immunology lab that I worked in at Southeastern. He never wore gloves. I'm like, dude, you're freaking crazy. He's like, epithelial scales, they just slough off. It's no big deal. I'm like, I want to have kids. You and your wife decided not to have kids. I want to have kids. I'm going to wear gloves. Here on campus, they won't let us bring it on campus. They won't want to deal with the biohazard of aethenium bromide. So there's other safe alternatives out there, one of which I used in the workshop and that we'll utilize in our biotech and biology and microbiology programs for visualizing DNA in the gel. But that's why I picked this particular mutagen to talk about, because, it's been widely used in research, and, we've known for a long time that obviously it could cause mutations, and so we exercise caution when using it, and have to be safe about the disposal of it. The other groups used the aethenium bromide. My group got to use the safe stuff. We get to try out the new stuff. They have this little PCR machine, it's this big, that you can buy in this little gel box that we use that's this big, and you can run it off your cell phone or your computer. So cool. $900 though. That's at a discount. So what else happens? So with radiation, there's different types. UV light, and there's X-rays, right? And X-rays is the worst because you're literally having electrons thrown at you. This is referred to as ionizing radiation. And so it can actually break the DNA backbone, and it's really bad if it breaks both strands, right? Just like cuts the staircase. If one railing gets cut, you could kind of repair that, but imagine cutting both railings of the staircase. You just fall. UV light, if you have two thymines that are next to each other, instead of hydrogen bonding with the adenines across the strand from them, right, their little intimate handshake, instead they covalently bond with each other next to each other. Covalent bond is like a serious hug here, peeps. Because what are they sharing when they covalently bond? Their electrons are shared. Right? This is a much stronger bond. This is a hug, baby. This is not a handshake where you could break away quickly. We're talking full-on hugging each other. Right? So when they do that, look what happens to the... You want to share or no?
Actually, it's the uncomfortable hug that you can't have.
Oh, yeah, that too. Okay, so you get this buckle, right, that happens in the sugar phosphate backbone. So again, remember that sugar phosphate backbone is really important for that enzyme sliding down. So now you got the bump in the railing, right? So what happens to the enzyme when it gets to this bump? Falls off or stops. Either way, is that good? No, because usually you're going to want to read this information, right? You don't just have it there just to be there. You have it to be read and used. So can you fix this? Yes. Right? And this brings us into, well, how do we fix these problems? Right? So you need to be able to identify the enzymes that have the ability to proofread DNA as it's being replicated. Well, this is kind of a no-brainer. Anybody know what makes DNA? DNA polymerase. So guess what? DNA polymerase not only makes DNA, but it proofreads it. So just like you could essentially proofread your own notes, right? Do you always catch your mistakes? Nah. Does DNA polymerase always catch his mistakes? Nah. Sometimes they slip through. But sometimes they don't, right? Sometimes you fix your own mistakes. So then we're going to look at the molecular level, right, at this DNA level, what excision repair does direct, mismatch, and SOS response. And among those, we really need to, not only understand, okay, excision repair does this, but why would you do that? What type of mutations are you trying to fix when you're excising, cutting out pieces of DNA and replacing it? And then we have this other problem too, is like, do they always mess up both sides? No, that's not how DNA replicates, does it, right? It splits in half, and one strand is used as a template while they're making a copy of the other strand. So one strand is the template, or what you would say is the old stuff, right? The original information, and then the other strand is the new. And of course, where does the mistake happen? Old or new? In the new strand. How does the cell know which one is which? Methyl group. It tags it, right? As your DNA matures, it gets methylated. Carbon and hydrogen atoms attached to it. So the old DNA is labeled, it's marked with methyl groups. So the enzymes know, okay, that's the template. The new stuff hasn't been methylated yet, so it can recognize that it's the new, and that's where the correction needs to be made. So we already answered that one, right? DNA polymerase are able to excise incorrect bases and replace them, right? They have their own proofreading ability. They not only make DNA, but they can check their work and fix it. In excision repair, this method is typically used when there's distortions in the DNA. So in the structure, right? The spiral staircase is not beautiful. It's distorted. So what type of mutations, one of them we just talked about, could cause a distortion in the structure of the DNA? Right, the thymine dimers that form that covalent bond with what type of radiation? UV light. So one of the ways we'll fix that is we'll cut out that section and we'll replace it. Now there's one I talked about way back, base substitutions, that could cause a distortion. It has a particular name, but in general, what could go wrong with base substitutions? Why would that cause a distortion maybe, right, where the structure isn't perfect? Talking about base substitutions. Yeah, double ring with a double ring, or so a purine with a purine, or a pyrimidine with a pyrimidine, where it's supposed to be purine and pyrimidine. We're supposed to have 3 rings across, but instead we end up with maybe 2 or 4, right? That would create a distortion, right? So those types of base substitutions where you're switching the different types of nucleotides. You're not staying within the same group. So this is just showing, the wrong one got put there, and they're excising it out and putting the correct one there. Here's excision repair for UV light. So here you see the 2 thymines supposed to be binding across, hydrogen bonding with the adenine. They're not. They're sharing electrons. They're in a serious hug here, and they've got the buckle there, right, in the sugar phosphate backbone. So the enzymes recognize this. They cut this out, and then they remake a new section. The next is direct repair. And this is sometimes referred to as light repair or photoreactive repair. And the reason for that, unlike excision repair, we're not going to cut anything out. The enzymes clasp onto this region, and using light energy, they break that bond between the 2 thymines. so that they go back to hydrogen bonding with each other. And notice this is mature DNA. Both strands have been methylated, right? This isn't a problem, you know, where they put the wrong bases there or anything like that. This is a problem that these guys have gotten a little too intimate, and we need to break it up. So direct repair is for thymine dimers, and also guanine can sometimes be damaged. So again, the enzyme, why it's called direct, it directly attaches. And then with light energy, which is why sometimes it's called photoreactive or light repair, that energy is used to break the bond. So if you have an organism that, say, you blast with UV light, but then you leave it exposed to natural light, could it repair itself? Yeah, if it has this direct repair mechanism, right? But say it's got this repair mechanism, we blast it with UV light and then we stick it in the dark. Is it going to repair itself? No, because it needs the light energy for this to work. So some people are like, well, why would you have excision repair and light repair? Well, because is it always light out? No, you want a backup, right? So that if you can't fix the UV light with light repair, you can do excision repair in the dark, right? Because it doesn't need light energy. You could do it in the light too, right? It doesn't require light energy, right? Excision repair can happen whether you're in the light or the dark. It's going to cut out that section and replace it. But if you have light repair, right, direct repair, you've got to have light to do it. Mm-hmm. Yep. So again, the enzyme directly attaches to the guanine that's been damaged, and it uses light energy to fix that damage. So mismatch repair. What type of mutation are you seeing here? A base substitution, correct. Right? One base has been switched out for another. What is supposed to be here? Supposed to be a C, but they stuck a T there, right? DNA polymerase screwed up, and he didn't catch his own mistake. So these other set of enzymes come along, right? And they recognize, oop, mistake, better cut it out. So these are similar enzymes to excision repair because they're going to cut out this section, and they're going to replace it. But they didn't cut it out because it was distorted, right? Like thymine dimers. They cut it out because it wasn't matching. So these enzymes are reading, right? They're not looking for distortions. They're reading. They're looking for these mismatches. Now, how do they know which side to cut? Right? The methylated, we know, is the template strand. It's only proofreading the new strand, which is not methylated. And that's the one they'll cut out and replace if there's a mismatch. Hence why it's called mismatched repair. So this will help with base substitutions if they're not caught by DNA polymerase itself. The last is when DNA damage is severe. DNA replication is going to stop, right? If there's a lot of distortions, right, there's a lot of mismatch, DNA polymerase is like, oh, I just can't do it. I'm just going to quit. It's too much, right? It's too much for me to handle. So then the system's like, oh no, DNA polymerase just quit. What are we going to do? Well, we're going to hire someone else who will do it no matter what, right? Someone who's desperate. So we're going to call up Mr. DNA polymerase that will just keep on going, keep on trucking, and he'll just do the best he can. Like, okay, it's, you know, it's stuck together here, so I'm just going to throw 3 T's here, and I don't know what that is, but I'm just going to put a G there, right? Because he doesn't care. The job is just copy, right? No one said I had to do it right, right? You know, you're not paying me but peanuts, so, you know, I'm just going to do what you told me to do. You just said copy. You didn't say Do it right. Or so what if you said do it right? I'm just going to do it this way. What does SOS stand for? Save our ship. Right? So, we're trying to save the ship, right? Because it's going down. Like DNA polymerase, regular old DNA, he just quit. So in order to keep up our line, right, to keep our line going, right, this bacteria is like, I got to make babies. I got to copy my DNA to make babies. It's just got to be done. You can't quit. Someone else has got to step up, right? So they get this other DNA polymerase that's not picky. You know, he's not all about perfection. He's like, I'm just going to copy it. I'm going to do the best I can. So he's actually going to definitely make mistakes, right? The DNA screwed up. But would you rather mistakes or die? Right? Hence why you say, please save my ship, right? I don't want to die. Please save us. So this is SOS, last ditch attempt, right? When the damage is so bad, right, that regular old DNA polymerase says, I quit. So then you got to hire the cheap guy. And he comes on board and he does the best he can to keep that ship from going down. Right? It might still go down. You might survive, but boy, oh boy, you're going to have some serious duct tape in the engine room. Right? Make sense? Okay. So, mutations provide organisms a way to respond to changing environments. It is, in fact, the environment that selects for the cells that are suited to survive. So the cells are always changing, right? They're always mutating. But it's that selective pressure of the environment that determines who's going to survive, right? We know this is natural selection or survival of the fittest, right? It is not necessarily the environment that causes the mutation, right? Mutation happens. It's the environment that determines whether that mutation is good, bad, ugly, or okay. Does that make sense to you guys? Right? Because some people get confused and they think the environment causes the mutation. But in fact, mutation happens. The environment determines what is good, bad, ugly, okay, as far as mutation goes. So everyone's heard about the peppered moths, right? I'm like so over the peppered moth. You know? No? Okay. Even though I'm over it, I'll tell you guys about it. So there's these moths. I forget where in the country this happened. And they had little black specks on them. So they were white moths with little black specks. So that's why they were called peppered moths. And they lived on the birch trees. And the birch trees are like that. You know, they're kind of whitish with like black spots. So they blended into the environment, right? They'd land on the tree. Birds would come by, fly right past them, wouldn't even eat them. Some of them, sometimes, due to mutation, would be completely black. They weren't spotted, but they were black. So when they landed on the tree, bird came by and went, thank you, I'll take that. Because they didn't blend in, right? So they were rare because they'd get eaten, right? The environment, under those conditions, they wouldn't survive. Well, then the area became industrialized, right? And whatever they were producing, they were putting soot in the air, right? Ash. And this was landing on the trees. So then the trees, instead of being white, were now black. So the peppered moths that were black now blended in, right? And so they're on the tree, and they're camouflaged. Their other buddies that are white with black spots got eaten up by the birds. So now you mostly have black peppered moths instead of peppered moths. See? The environment determined who was going to live. The mutation was always there, but we got that shift in the population because the environment changed. Of course, the people were the ones who changed that, by the way. Right? In my lab class last night, they were talking about, you know, how sometimes you can't go swimming because of the bacteria. Well, some of that is environmental because, like, it hasn't rained in a long time. That would normally flush the bacteria out of that area where you're swimming. Some of the algal blooms and stuff that we deal with, it's because we put too much nutrients in the water and then they overgrow. So, I mean, It's not the algae fault, it's our fault. We gave them too much food, right? And this can happen too with other things, right? It's a natural phenomenon, but it gets exacerbated because of stuff that we do on a large scale, right? Okay, I can get through direct and indirect, but I don't think we're going to get through gene transfers, but I'll add them into our activity. So please go over that material, right, before the next class. And the animations, definitely, right, the bonus, if you haven't done them yet. I think those are in that packet. There's a lot of videos for the 3 gene transfers. So the next thing, and actually this was brought up at the workshop that I was at, they were like, well, why don't you do replica plating, right? And some people actually do this technique for the Small World Initiative. But again, when you've got to deal with, you know, certain resources, certain materials, not everybody has access to these things. So direct would be really easy and simple, right? Because you could directly select for the mutant you're looking for. Well, that's great if you know what the mutation is, right, that you're looking at. So these guys are just colored for understanding, but all these bacteria in this tube, they would all look exactly the same normally. But different ones have different characteristics that we could test for. So for instance, especially with Small World Initiative, we're looking for bacteria that produce antibiotics, right? So we take those bacteria and we expose them to other bacteria and see if they can kill the other bacteria. So then we know that bacteria is producing antibiotics. So then we take it and we isolate it so that we've got that single species of bacteria, and then we grow it up and we isolate the antibiotic that it's producing from it. Well, here they're using antibiotics to actually select for ones that are resistant to antibiotics. So here you have cells, some of them have become resistant. So if you grow them in the presence of the antibiotic, say streptomycin, on this plate that contains streptomycin, only the mutant that's resistant to streptomycin is going to grow. And that's great. That's your mutant. You pick it off the plate and you go study it, right? But if you don't have that selective pressure, again, this is just colored to let you guys know. These would not be colored. All these guys would look exactly the same. You wouldn't be able to tell one from the other. Still with me? So, but they're directly selecting here by killing off all the guys that are sensitive and just leaving behind the resistant. That's great if you know that that's what you're looking for. It's not always the case. So indirect selection is sometimes necessary. And in this case, you would have colonies on a plate and you would use sterile felt. You know what? Felt is kind of fuzzy, has little fibers, right? So you basically are using it to create a stamp. So you would have a line on both plates and you would stamp it on. So the felt would pick up little cells from each one of the colonies in the place where they are on the plate. And then you would, again, lining it up, stamp it on different plates. So different media, you could say, right? And we do this for Small World because sometimes, you know, I mean, these are unknown bacteria. So we don't know what they like to eat. So you could put them on one media and they don't grow, they die because it doesn't have the nutrients they need. And then you put them on another one and they grow really well. But how are you going to tell one colony from another? Like, are all these guys really the same? Well, they look the same on the plate, but under different environmental conditions, they might behave differently. They might not even grow. So if you stamp this on 2 different media, and then you compare these 2 plates, you notice, oh, this guy right here isn't on this other plate. He's dead. So this media right here doesn't contain something he needed. He's mutated. He can't do this on his own anymore. But we didn't kill him in the process. We saved him. He's over here, right? So we could pick him off and study him, right? We know he's a mutant. We know he can't do this, whatever it is in this media, right? So let's figure out what it is. Let's take him and study him further. So they do this with the, you know, except instead of felt, they have this special square thing that has all these pegs, and what you do is you stamp it on, you actually use 96 well plates, liquid culture. So each little one is a different test. And then you could pick up the bacteria from those 96 wells with this like stamp-like thing that has these little prongs, so it picks up a little bit of each one, and then you stamp it onto a plate, and it's a square plate too. And you make sure you orient it the same, so then you can study that same organism very rapidly, easily on different media by just stamping. So hopefully he'll send me his PowerPoint and I can add that stamp thing to your lectures. I didn't get to see it in action. We didn't do that. But, this is the cool thing of going to workshops and everybody comes from everywhere. They share what they do, where they're at. We even video conference with people all over the United States too. It's crazy what you can do with technology now, huh? It blows my mind. Does this make sense to you guys, and why you would want or need to do indirect versus direct when you're trying to find your mutants? So the Ames test is kind of like this, where we're studying mutation, and when you have chemicals, like say a company comes up with a new antibiotic even, right? One of the things that they then do is actually test it on eukaryotic cells, like maybe plant cells or yeast cells or something like that. A lot of times too, like if a chemist develops a new chemical, they're going to test it on bacteria. They're going to do the Ames test. And what the Ames test does, it actually starts with a mutant that when it mutates, it reverts back to the form that can survive without histamine. So these mutants will grow up if they've been mutated. If they don't get mutated by the chemical, they don't grow. And then, of course, you'll have a control, right, and they don't grow. But if the chemical mutates them, they'll grow under these conditions, right? They'll revert back, and they'll be able to grow without the histamine, amino acid added to the media. So in this case, this means that that chemical changed the DNA of those cells. So would you want to use that chemical? Would you approve it if you were the FDA? Certainly not, right? Because we know mutation can lead to cancer, right? We don't want anything screwing with our DNA, especially not in a way we don't understand or know. So next time, as I said, I'm going to try and work these into our activity. So we're done for time. 5 minutes sooner than I wanted to be. So that's good.